You need to be able to describe and explain the effect of temperature on an enzyme's rate of reaction. This is a little bit of a link to chemistry and rates of reaction in chemistry. Because as temperature increases, the particles, the substrate and the enzyme will move around more. There will be more collisions between these two, so a reaction is more likely to take place. The enzyme is more likely to meet the substrate and form an enzyme-substrate complex. We can see that on a graph with temperature across the bottom and rate of reaction up the side. There are two parts to this graph. In pink, we can see what is happening as the temperature increases, but that is not the whole story. Because as the temperature rises, the bonding within the enzyme starts to break down.
This causes a change in the tertiary structure, which changes the shape of the active site. This means the shape of the active site is no longer complementary to the substrate, so no ES complexes can form. This causes the rate of reaction to decrease. As you can see on my graph, it has gone rapidly down, and we say the enzyme has denatured. It has not been destroyed and it has not been killed because it is not alive. It has been denatured. The optimum temperature of an enzyme can be found at the peak of graphs that look like this, with temperature on the x-axis. Different enzymes will have different optimum temperatures. Human enzymes that work inside the body will have an optimum temperature of 37 degrees C, because that is body temperature. We need to know about the temperature coefficient, often referred to as Q10. The temperature coefficient is the factor by which the rate of a reaction increases for every 10 degrees centigrade rise in temperature. We can calculate Q10 using this formula. It's the rate of reaction at whatever temperature it is currently plus 10 degrees C divided by the rate of reaction at whatever the temperature it currently is. For chemical reactions carried out in a test tube in the lab, or for a metabolic enzyme-controlled reaction between 0 and 40 degrees, its optimum temperature, we're assuming these enzymes are human enzymes from the body, the Q10 is normally around 2. This means that for every 10 degrees increase in temperature, the rate doubles. For enzyme reactions that are happening above their optimum temperature, the Q10 will drop because the rate of reaction is going to decrease due to the denaturing of enzymes.